The polymerase chain reaction, or PCR, is a molecular biology technique for amplifying a piece of DNA into several millions of copies. The technique has several cycles of heating and cooling of the reaction for DNA melting and enzymatic replication of the DNA. A thermal cycler is used to carry out cycles of heating and cooling necessary for carrying out a PCR. The components for PCR includes DNA template, a set of forward and reverse primers that specify the target region for amplification, a heat-stable DNA polymerase enzyme, named Taq polymerase, and nucleotides necessary for synthesizing new strands. First, the temperature is raised near to boiling to separate the double-stranded template into single strands. Then, the temperature is reduced to allow annealing of the PCR primers to their complementary regions on the template DNA. At a higher temperature of 72 degrees Celsius, the enzyme Taq polymerase binds to the primed sequences and add nucleotides to synthesize the second strand. The enzyme reads the sequence on the template to add complementary nucleotides. Adenine complements thymine, and guanine complements cytosine, or vice versa. This completes the first cycle. This process of denaturation, annealing and extension are repeated in several cycles to make many copies of the target DNA. The number of target copies increases exponentially with each cycle of PCR. Number of cycles determines the final yield of target copies. PCR is widely used for enrichment of DNA fragments for a wide variety of applications, such as gene sequencing, genetic testing, detection of pathogens, forensics, DNA fingerprinting, cloning, etc.